Hi, I'm Ann Arneson, and I worked in Dr. Dan Gary's lab this summer. My project was to develop an in vitro system to screen human-induced pluripotent stem cells for their compatibility with porcine parthenogenetic embryos. HIPS cells hold a lot of potential in the field of regenerative medicine, but they're highly variable in nature. So the aim of my study was to determine if porcine parthenoids could be used as an experimental model to test this potential. Long term, this system could be used to grow human organs in pigs for transplant or to study as models of human disease.
Hi, I'm Elizabeth Loxo, and this summer I worked in Dr. Paul Izio's lab. We were interested in investigating how a swine heart changes when you take it from in situ, which is in the body, to in vitro, which is the visible heart apparatus in this case. We measured the mitral valve, aortic valve, annulus areas, the wall thicknesses, and the ejection fractions. This work is important because it can help us better design medical devices.
Hi, my name is Emily Ruin and I worked in Dr. Thomas's lab this summer. I studied an enzyme called SERCA, which transports calcium from the cytosol into the sarcoplasmic reticulum of muscle cells to cause muscles to relax. In heart failure, SERCA stops working. Our lab does drug screening to discover small molecule activators of the pump to improve its function. Once we find those activators, we want to characterize them in a controlled environment. So I tested two different methods of protein isolation: homogeneous preparation and microsome preparation. Through Western blots, enzyme activity assays, and distance measurements of the protein using fluorescent probes, we concluded that the microsome preparation offers us the best platform for studying future SERCA activators to treat heart failure.
Hi, my name is Emily Sivrud, and this summer I worked with Dr. Paul Izio in the Visible Heart Lab. My project focused on designing a new protocol to MRI scan perfusion-fixed heart, lung, and blood specimens. I designed 3 prototypes to tether the lungs and maintain them in the attitudinally correct position during imaging. Once I obtained clear scans, I imported them into Mimics, which is a 3D modeling software, to create detailed and correct models that we could then print with our 3D printers. The models, once they're printed, can be used for educational purposes in the medical school or for surgeons as they plan to design new surgical protocols.
Hi, I'm Carly Bowl, and I worked in Dr. Michael Kaiva's lab this summer. My research looked at a developmentally relevant transcription factor called MES1, and we found that when MES1 is induced in mouse embryonic stem cells, it produces a cell progenitor population that has dual cardiac and myogenic potential, meaning it can make both cardiac and skeletal muscles. So this is important because it is similar to a part of the embryo called the cardiopharyngeal mesoderm, which makes the secondary heart field and the craniofacial muscles, both skeletal and cardiac muscle. So this means that we can use this knowledge to study this part of the embryo and in turn study development of the heart. And this information can be applied in many ways, such as understanding congenital heart disorders or applying regenerative techniques in medicine. Hi, I'm Megan, and I worked in Dr. Don Lau's lab this summer. My project was studying the effect that estradiol and progesterone has on cardiac muscle in mice. We had 5 different treatment groups that would simulate different groups of postmenopausal women given different hormone treatments, and I tested 24 genes that were related to the inflammatory response, myogenesis, and oxidative stress, and using a real-time quantitative PCR. And this is important in understanding new treatments that would help reduce recovery time in postmenopausal women who have suffered a heart injury or a heart attack.
Hi, my name is Naveen Haragu, and this summer I worked in the Uravasti lab. The Uravasti lab studies Duchenne muscular dystrophy, a disease which is caused by the absence of the protein dystrophin in muscle cells. Dystrophin is a protein which helps link the inside of muscle cells to the extracellular matrix through a complex of glycoproteins in the cell membrane known as the dystrophin-glycoprotein complex. In the past, it has been shown that microtubule lattice organization in skeletal muscle cells is mediated by dystrophin. What we studied was looking at proteins which may mediate that indirect interaction between dystrophin and the microtubule lattice. In order to do that, we first performed an eye-track proteomic scan, which helped analyze differentially expressed proteins in samples from muscle cells with normal microtubule lattice organization and samples from muscles with disorganized microtubule lattice organization. Once several proteins were identified, we then used Western blotting to validate those results. Finding novel proteins which mediate this interaction may lead to novel therapies for patients with Duchenne muscular dystrophy in the future.
Hi, I'm Nicole and this summer I worked in Dr. Mary Gary's lab. My project was a pilot study that looked at the effect of novel newt extract on mouse cells. It is commonly known that newts and salamanders can regenerate their limbs following an amputation, and they do this by de-differentiating their cells into a stem cell-like state. Mammalian cells cannot naturally do this, but we wanted to see if we applied newt proteins to the mouse cells, if they could de-differentiate their cells in the same way. Our results suggest that there is a possibility that these mouse cells can de-differentiate when new proteins are added to them. This is an important experiment for regenerative medicine because if we could regenerate tissue like the newts do, we could make new tissue or even whole organs for patients that need them.
Hi, my name is Tanya Moore, and this summer I worked in Dr. Joseph Mester's lab. And what I focused on was understanding the function of proline because we thought that the proline and the platypus was used as a bridge from lower vertebrates to mammals. And this is important to the fact that we are studying how mutations in the heart can enhance cardiac performance.
Hi, my name is Scarlett Guo, and this summer I worked in Dr. Tranquillo's lab in optimizing magnetically aligned fibrin gels for contact-guided studies. We hypothesized that slower gelation rates would correlate to greater magnetic fibral alignment. This is important because cell contact guidance on aligned fibrin gels is the foundation for myocardial equivalents, which have been found to have greater stiffness and strength than neocardiac tissue, which are characteristics that we would like to eventually incorporate into heart tissue replacements for cardiovascular arrest victims.
Hi, my name is Tracy Den, and this summer I worked in Dr. Parlangero's lab, whose lab focuses on muscle development in an embryo. One transcription factor that's important in muscle formation is PAX3 because it activates the myogenic program. Two proteins that are thought to interact with this transcription factor are LDB1 and 6-4. So we would like to isolate these two proteins for analysis. However, current antibodies are not able to extract them. So the goal of my project was to tag these two genes with a flag tag in order to isolate them for analysis. I did this by using the Cas9 CRISPR system in mouse embryonic stem cells. This work is important because once we're able to isolate these two proteins, the interaction between the three can be better understood and muscle formation as a whole can be further looked into.
My name is Yuna Jung, and I worked in the Thomas lab this summer. And I worked on the protein dystrophin. Dystrophin is a muscle protein that helps relieve the force produced when a muscle cell contracts. When it's mutated, it can't relieve the force anymore, and the muscle cell tends to rupture. This causes Becker's muscular dystrophy. But despite the severity of this disease, very little is known about how the dystrophin molecule functions in the body. Therefore, this summer I studied the structural mechanisms underlying how dystrophin opens and closes. We will use the model we have discovered as a reference point to study the mutated version of dystrophin in the future. Hopefully, this will lead to a cure for Becker's muscular dystrophy.